Well, the question now is on whether gene editing will soon or finally become a reality. And I think there are great hopes and great expectations that we can find functional cures and probably intervening through genetic strategies could be the final frontier. And I think Joachim Hauber is about to tell us on their plans on getting to do that.
Yeah, thank you. Hello, everybody, and thank you very much, chairpersons, the Spanish AIDS Society, for the invitation. I'm happy to be here in this wonderful city. It's really, for us Germans, it's really something different. I mean, this guy comes from the southern part of Germany. It's warm there. Yeah. I'm from Hamburg. That's what we call freezing cold here. So by the way, so I'm from the Heinrich Penn Institute. It's the institute here, and actually Frank taught us how mankind acquired HIV. I want to now look in the other direction with you, how we maybe, if we are lucky in the future, can get rid of the virus. Okay, I got frequently asked why are we in cure research, and we don't have to go all through these points. And we fully agree that combination antiretroviral therapy is one of the big successes of biomedical research over the last 20 years and so on. Nevertheless, there are some downsides connected with it, and one is it's a lifelong treatment, and the longer you treat the patient, we see more and more so-called secondary toxicities like lymphomas, kidney disease, diabetes, and so on. And most of all, art cannot cure HIV infection. And if you talk to patients, they really, even if they control their viremia perfectly well, they want to get rid of the virus. This disease goes to bed with them, gets up with them in the morning, and they just want to get rid of it. And that's why why we should do cure research, basically. So the problem, okay, we have to distinguish about, for the non-experts in the field, there are two ways of cure. One was already mentioned by Jose, which is called a functional cure, or much better expression is long-term remission. So the patient is still infected with HIV, but controls his or her viremia perfectly well. So the immune system is like in this group of elite controllers controlling the virus. Patient is not infectious. Nevertheless, we can isolate replication-competent virus or pro-viral DNA from these patients. And the holy grail certainly is a sterilizing cure where we really get rid of everything in the patient, that the patient is fully cured and eradicated from HIV. To make this clear at this point, whether we will ever achieve a sterilizing cure, I cannot tell you at the moment. So this is the hope, this is what we should aim for. Nevertheless, if we are able to achieve a functional cure over the next years, and I'm fully convinced about that we will be able to do this, this would be just spectacular for the patients. Okay, what's the problem? The problem, as you know, is that HIV integrates its pro-viral DNA, establishing a pro-virus in host cells, and very frequently these are also latent infections. The main reservoirs are central memory and transitional memory T cells, but certainly there are also other reservoirs. But what a big caveat is, what became clear by Silesiano's work over the last years, is that there is a large amount of integrated pro-viral DNA that is encoding fully replication competent virus, but cannot be activated, or in other words, purged. So a shock and kill therapy with latency reversing agents is constantly failing on this reservoir of pro-viral DNA. So I think it's fair to assume that any eradication attempt will greatly benefit from a technology that directly targets the pro-virus or the pro-viral DNA. I'm personally convinced, without going after the pro-viral DNA, this is not everything, it's not a silver bullet, but without it, there will be no cure at all. So for a couple of years, we have the tools to go after the pro-viral DNA, and this is listed here. Homing endonucleases, you know them, zinc finger nucleases, talines, RNA-guided nucleases like CRISPR-Cas and tyrosine-type site-specific recombinases. All these tools have ups and downs, have pros and cons. It's better here? I don't know. Certainly, and if you are more interested, then we can discuss today. I just referred to our most recent review and current opinion of virology, where you can read up on all these technologies. Here, for the sake of time, I want to concentrate certainly on CRISPR-Cas and on tyrosine site-specific recombinases for eradication, excision, inactivation of HIV proviral DNA. So let's start with CRISPR-Cas because it's such a hype, and I have to admit we use this every day in the lab. It's such a great enzyme system to inactivate genes. Why? Because it's easily targetable. So the nuclease by itself is not recognizing the target sequence. It's rather recruited by a short RNA, which is called guide RNA, as you know. And basically every student can create this guide RNA within a week, or just ordering an oligonucleotide from a vendor. And when the oligonucleotide arrives, he clones it into his expression vector and can start to knock out genes or maybe HIV. So it's really easy to program. It takes days, basically. But there's also a big caveat, particularly if we talk about application in humans. And this is that the system, the mode of action of CRISPR-Cas and this type of designer nucleases relies on the cellular error-prone DNA repair pathway. And this we will see now. What's another problem is certainly also the problem of delivery. How do we get the system where we want it to have it? Because we have not only deliver quite big nuclease, but also multiple short guide RNAs expressed from different pol-free promoters. And this is always a problem to put this all together in the same vector and so on. So there was a series of publications actually this year employing CRISPR-Cas and looking for inactivation, excision of HIV-1. And between all these publications you see here at the bottom of my slide, they tested every guide RNA so far published in the literature targeting HIV-1. And this is just one example here. You see the, sorry, I have to go, what is this? You have to back. Okay. Let's start here. You see the different guide RNAs, so targeting in the LTRD theta box, another one, the TAR region, the RRE, for example. And what you see here, this is basically the P24 over here, release. You see the wild type, the untargeted virus is replicating. You see within a week, perfect inhibition, and you see breakthrough of the virus for every guide RNA. Very efficiently here, breakthrough. So from another study here, they used the guide RNAs also targeting the LTR, so in theory you should be able also to excise the virus. Just the same experiment or the same result. There's the wild type replication. You see the guide RNA, you see about a week, perfect inhibition, and then the virus comes up, is fully resistant. When they did next generation sequencing here, you see what happens. You see this at the target site, this what's called indels, insertions and deletions. The short deletions are short insertion. And very frequently, basically, you get this inactivation. Very frequently, just a few nucleotides are incorporated, which are not destroying the reading frame, but now the target site is not recognizable by the same guide RNA anymore. So it's not virus replication or anything like that. It's the mode of action of the CRISPR-Cas system forming these indels. Now, when you do this in the lab, you want to inactivate a given gene. Well, no problem, because you can select for your product. This is, you cannot select in a human body for the product. So you will create very efficiently, or even you will facilitate the creation of resistant virus. So as the system is a perfect system, you have to think twice whether you want to apply it in humans, particularly when you want to treat a highly replicating system like a virus infection. So the other system is around actually for many years, is the Cree recombinase, and I'm also sure many of you use Cree recombinase in your lab. It's a naturally occurring recombinase from bacteriophages, from the phage P1, and the target site is called LOXP. So what is the advantage or what's the disadvantage? The disadvantage, it's a pain to reprogram Cree recombinase, that now the new recombinase, the designer recombinase, is recognizing a new target sequence. It takes months, if not years. We will see in a minute a method called molecular evolution. But there's also a big advantage. It acts error-free, so it does not rely on cellular DNA repair pathway. So there are actually or literally no mistakes upon recognition or recombination in the DNA. And it requires rather simple delivery technology. You have only to deliver one single protein, which can be done easily with any kind of viral vector technology. So recombinase is also not only excise, depends on the orientation of the target sequences, of the flanking sequence. It can also translocate or inverse the sequence, or can be used for cassette exchange, similar like CRISPR-Cas. So now many labs for many years tried to mutate Cree recombinase, that now the recombinase is recognizing native HIV sequences, and they all failed. And the trick was, so when you mutate Cree and you want to introduce the mutation, you hope that it's recognizing HIV sequence. Originally you start with the LOXP sequence, you want to recognize an LTR sequence in HIV, it always fails. So the trick was to subdivide your target sequence and to raise for every target sequence a recombinase intermediate. This is shown here. And these intermediates are then combined to form the final product, the final recombinase. So in real life, this looks like this, and this is this really laborsome procedure. This took about 1.5 years here. We started here out with 6 subsequences of the target sequence, then collapsing this to the single recombinase here. So a little bit hardcore molecular biology. One can do this in pools, in bacteria. So for example, you have a vector, harbors 2 copies of your target site. Here the LOX LTR sequence of HIV, and these 2 target sites are flanking a single cargo, in our case here NDE1. On the same plasmid, you ligate in a mutated library of recombinase, for example, and now the entire pool goes into bacteria. So the bacteria express the recombinase and they work on their own plasmid. So commonly, nothing will happen. There will be no recombination. However, in a few cases, the recombinase will take out, by recombining these 2 target sites, the single cutter NDE1. So the next day, you can isolate the entire pool of plasmids, treat it with NDE1, you get rid, you linearize all what you are not interested in, and now by PCR, indicated by these small arrows, you can rescue your library, and you can do this in cycles and cycles. It is known as molecular evolution. So what we came up with is a recombinase now called BRAC, Broad Range Anti-HIV Recombinase 1, which recognizes the vast majority of all HIV clinical isolates. So you see the target sequence. This was the original top line from Cree, our previous Cree recombinase, and also now the new target sequence, which is located in the LOX, in the LTR region. Is my time up? Oh. Okay. What you see here, when you went in the Los Alamos database, that this sequence is preserved in 90% of all HIV isolates. And when we go into the clinical relevant isolates, at least here in Europe and North America, subtype B can treat about 94% of all HIV with this single recombinase. So as a matter of fact, in theory, we can treat 28 million HIV-infected persons from the currently 36 million infected on this planet. So is this recombinase active? in mammalian cells, we transfected here HeLa cells with a reporter construct flanked by recombinase HIV recognition sites. So when the recombinase is active, it takes out the mCherry reporter, and then the GFP gene gets activated by the promoter, and the cells turn green. You see the BRAC recombinase on its homologous LTR region target site. You see it's active in all of these cells. It's not active on another sequence in the LTR or on the LXP site. And these are the controls of Cre recombinase. And it's highly active in mammalian cells. So what happens then? The recombinase, or what happens in the cell, the recombinase recognizes the flanking LTRs, excises the sequences in between, and forms a transient circular recombination product. This recombination product cannot reintegrate anymore, and it's degraded by nuclear or cellular nucleases over time. So you take out all HIV genes from the site of integration. And this you can visualize directly, basically, because you can use specific primers for PCR, and you see a cell culture expressing via lentiviral vector the BRAC recombinase. You see at day 1 after transfection, you see how the excision product comes up and goes down because the culture is then free of HIV. And visualize this as P24 release as the control, and after about 10 weeks, it's completely free, no HIV in there anymore. So the nice thing is here that you can use this for sequencing, and you see here, this is the target site. Every nucleotide in the target site is preserved on the excision product. So there are no indel formation. So there's a high level of biosafety if we talk about gene therapy for HIV. So what are the side effects? We did some bioinformatics and went to the human sequences 1 to 6 that resemble most closely the HIV target site. So the mismatches are again indicated in red, and here down you see we tested them here in the lab. None of these sequences is recognized, so it's highly specific. And we did all these genotypic assays like SCI assays, genome arrays, and so on. We also looked into immune activation in primary cells, or on using clonogenic assays on stem cells, cytokine release in primary cells, and so on. So there is no cytotoxicity or genotoxicity observable, even upon months of expression. We even generated transgenic mice expressing the hematolymphoid system constitutively the recombinase, and the mice are no different than their offspring, and the offspring is perfectly, and it's mating, and everything is fine. So now we have to talk, how can we apply this to humans? There are basically in gene therapy 2 ways. You take out the peripheral blood, the T cells, genetically modify the cells and put it back into the same patient, or you mobilize hematopoietic stem cells, do the same and put it back in the same patients. And we have to look into both possibilities. So what we did first, we isolated from HIV patients PBMCs, cultured them for weeks, transduced them with a lentiviral vector expressing the recombinase, and then monitored viremia and also performed next generation sequencing. And this is the example here on top. You see the control from the patient. So the virus is replicating, goes down because the culture is going to die over time. This is after 20 weeks of transduction. You take the same cells and bring in the recombinase, you get a completely different picture. You see that always with the same or similar kinetics between week 15 and 20. You cannot detect virus anymore in this primary T cell culture from the patients, and the cells are perfectly protected. Now we can isolate the genomic DNA from both cultures, perform next generation sequencing, and there's an example here. These bars are the reads, the 500, 600 nucleotide reads of NGS, for example, chromosome 12. And this is before and after for HIV-specific sequences. So even if the culture is cured from HIV, you always will pick up some HIV sequences, as you have seen before, because many of the integrated proviruses are replication incompetent. These they will score here as the sequence. So then we have to test this in vivo, and what we have established are humanized mouse models, NSG or REC2 animals, that we either engraft or transplant with human primary T cells or human hematopoietic stem cells, either isolated from cord blood or peripheral stem cells from patients. So we can transduce them, we can engraft the animals, and then we can either infect them or we transplant them already with patient cells that are already infected, and we can monitor viremia over time. And this is shown here. This is the experiment, how we did it, basically. And this is one example again. You see control, you see viremia. These are now weeks after post-transplantation. This is the animal that received the recombinase. You see at the week 18 ongoing, no plasma viremia at all with the most sensitive assays. It's not just a single animal. There are 2 more HIV patients. Again, no viremia detectable anymore. And certainly we can also go into, or we can transplant the animal with stem cells, not from patients. This is for ethical reasons still not possible. But then we infect the animal with HIV. And this again is the control. Here are 2 animals here, have a little blip, another one below the detection limit. So we can isolate, rescue human cells from the animals, sacrifice the animals. These are 2 animals, and isolate whether there are still integrated GAG sequences from the proviral DNA. And see, this is the BRCA-treated animals. These are the control animals. We do histology, the spleen here for HIV-infected cells. You see the control animal. You see easily here the red dots, HIV-infected cells, and you really have to look hard to see any HIV-infected cell in the BRCA-treated animal. So I'm more or less at the end. So I showed you that BRCA can target an organ. This is, I think, also is a big advantage, completely independent of tropism or clade or whatsoever. You can target every HIV virus and established infection. You not only prevent infection, you treat an established infection. It's also important. It's highly precise. There are no cytopathic or off-target effects. Has a strong in vivo antiviral activity, and this now allows to go into a so-called phase 1b/2a study to test this in HIV-infected patients. So what we believe, I mentioned this already, an approach like BRCA recombinase or even some other antiviral gene therapy approach, this will never be the silver bullet that this one approach will work with respect to cure, but it will form an absolute essential basis in a combination cure therapy. So this is the idea. At the moment, most experts in the field believe that you have to target the hematopoietic system, hematolymphoid systems by modifying the hematopoietic stem cells in the bone marrow to provide a constant source of resistant HIV-resistant cells in the periphery. But we can conceive that basically in the beginning we can also add up to this with a T-cell transduction in the patient. And what we're really working for, it is a goal that's currently going on in the lab, that we use tissue-specific targeting for BRCA recombinase, for example, AAV vectors delivering BRCA1 in, for example, CCR7-specific cells, which basically constitute the majority of the HIV reservoir. Another example is that it can be easily combined with all other antiviral approaches. I prefer immune modulating strategies like broadly neutralizing antibodies or advanced shock and kill approaches. So at the moment we are in the process discussing this with the overseeing agencies in Germany, and we basically cleared the outline of the first initial study, and at the moment we are raising the money for this. that we go into HIV patients that also suffer from large B-cell lymphoma. And they have to undergo R-CHOP therapy, and this gives us a time frame to mobilize also the patients, take out by leukapheresis the hematopoietic stem cells, modify them, and reinfuse them back into the same patient. And the neat thing is these patients will always be on ART, so there's no risk for them, but we can follow up then the marking of the cells, the reservoir with our molecular methods, and if the patient agrees, we are allowed to take them off ART and very closely monitor whether there is a rebound or not. So this is what's in the pipeline for the next 2 years, more or less. And with this, I'm at the end. I want to acknowledge our collaborators. Ilona put up all these animal systems. Our colleagues in the lab are all experts in toxicity, vector development, and so on. We really closely work together, and it's really important with our on-site clinicians, Jan van Lunsen, Olaf Degen, these are the AIDS doctors there, and also with Nicolas Gerger, who is the bone marrow specialist. With Frank Buchholz and Janet. Janet was a former postdoc in our lab and then moved also back into Frank's lab. These are our collaborators, the experts on the nuclease technology. Our lentiviral vector we obtained from Axel Schambach from the medical school in Hauber. So that's where we are. This is the University Medical Center in the Hamburg-Eppendorf district, and the Heinrich Petty Institute is located here, right at the border. And with this is part of our new symphony hall in Hamburg. You have to see this. I thank you for your attention.
The talk is open for questions. Frank.
Very nice presentation. You mentioned that it's active against about 90% of all primary strains, which is very nice. But in people, you also have quasi-species. And I think the estimates are that you may have up to 1 million latent proviruses in a single person. Have you actually done deep sequencing analysis to check how frequently you already have variants that are maybe resistant against this approach in like regular people?
There's a big consideration, certainly. From our experience is that so far we haven't, we really looked hard to see whether there's a resistance development. We failed so far. And we did also some deep sequencing in patients, and all sequences we pulled out were recognized by the recombinase. We also mutated the sequence, the target sequence in LTR, and this is unpublished, but I can tell you some single nucleotide substitution are still recognized by the recombinase. So actually, to be honest, we are not at 94, we are at 97%. Yeah. So far, we couldn't detect anything. Certainly, we would do this before we treat the patient to do some deep sequencing. And if this would be a patient who harbors different viruses, he will not be treated.
Very nice. Can you just clarify the data that you were showing with the RNA that basically went down with the construct in the reactivation of the virus from patients that were HIV infected? Was that correct, that you took the cells and you modified the cells in vitro and then showed that the RNA went down as a consequence of
Release
protection?
of particle release.
Particle release.
Particle release, and
Correct.
then isolating genomic DNA and looking for integrated DNA.
So I'm looking at this data from the context of the estimate is 1 integrated latent copy per million latent CD4. And when you take the PBMC and you take the CD8s out and you reactivate a PMA-N-omycin, not every patient reactivates virus. But once you see it, then that obviously is a reflection of amplification of a latent pool. Now, your modification came in after the cells were making particles, correct? Because even the modified group had RNA-positive particles. So are you coming in after the reactivation as opposed to before the reactivation?
I mean, there are always limits to this. And your point is, certainly we cannot target every latently infected cell in a human being. Nobody can at the moment. Also with all other...
Sure.
Sure.
I'm just trying to see whether what you're doing is you're basically cleansing out the virus within in vitro, meaning you're modifying the cells to the point that any cell that becomes reinfected in vitro now is able to survive because it basically removes the integrated site.
Yeah, well, these were in pooled cultures. We not selected for transduced cells. So in the culture, there's still enough cells that are not protected because do not contain the lentiviral vector.
Right, and that was my second question. What's the penetrance? Because in the lentiviral systems, they talk about 5%, 10% penetrance overall. Do you see yours better?
Well, usually, on average, we have transduction rates of between 30%.
30%, so yeah, so that's better.
Something like this. But there are new methods in the pipeline. You can check up transduction rates up to 90%.
Because even in the zinc nuclease, they say, well, even if we don't protect every cell, the cytokines are protecting bystander cells. So there's all this dialogue about that
sure.
penetrance is not everything.
That's true.
But I didn't know if you thought that was also the case in your system as well.
Well, the in vivo data are really breathtaking because we really push this down below the limit of detection, and it's lasting. I didn't show you this. The mouse lives usually a year and never comes back up, the virus. So it's really lasting the effect. And I only can explain this, that there are some bystander effects, certainly. Yeah.
In the same line, do you think actually, because I think it somehow relates to the previous two questions, and it may come to an advantage, your first trial on patients receiving CHOP, meaning if you in fact induce immune suppression and destroyed potently latent infected cells, coming with a silencing technique would be more effective. And so this is more a comment than a question, but will that happen?
I just don't know, I have to admit.
Because I think the problem will be by targeting, how are you going to manage to target every single latently infected cell?
No, this is a problem of the entire cure field. The entire cure field. As I just mentioned, you never will reach the last infected cells. What you're hoping to do is, whatever you do, that you functionally reconstitute the immune system of the patient, and that a reconstituted immune system is finishing off the last cells. But then we are talking about sterilizing cure. Our first hope is to have a functional cure.
All right. Thank you very much.